It's with the deep sadness I present this brief tribute to honor the memory and contributions of late Professor Lindsay Black, a distinguished member of the PVA community. Lindsay Black was there from the start of the PVA, and his contributions to bacteriophage assembly spanned for more than 55 years. It was a shock to everyone when we learned of Lindsay's passage. Lindsay was one of the most fit persons I know. He was an avid biker and hiker and didn't seem to age at all and gave the impression that he will live forever. I still cannot believe that he is not with us today in the PVA conference. Lindsay had an extraordinary career. He graduated from University of Chicago, did his PhD in Stanford Biochemistry Department, then went on to postdoctoral research in Edward Kellenberger's laboratory at the University of Geneva. He then moved back to US and established his research program at the University of Maryland Medical School, where he remained for more than 40 years. Lindsay had participated virtually in every PVA conference, yet there are very few pictures of him. He was an extremely shy person. I did receive a few photos from colleagues. Let me share these with you. This, I believe, from his early days in Switzerland with Alastair Stephen. These are some of the pictures from past PVA conferences. Here is Lindsay with his wife, Judy. Here, a photo from Helsinki PVA in 2003. And this is actually from FASB virus assembly from Vermont with late Gisela Mosek. This is a PVA conference picture in Syria, Virginia in 1993. In fact, Lindsay and Alastair here organized this conference. I believe here is Fred Iserling. This is a picture from Finland conference in 2003. And this is a picture with me in Annecy, France, 2009 PVA conference. This is with some of his friends hiking in Virginia mountains in 2012. A memorial issue on phage assembly pathways is underway for the journal Viruses. Please contact the editors, Andreas Kuhn and Julie Thomas, if you are interested. I was one of the first postdocs with Lindsay, probably the longest time a postdoc spent with him, for about 9 years between 1980 to 1989. And I had the pleasure of reading probably every paper that Lindsay had written. He made many seminal contributions to bacteriophage assembly. I can only touch upon some of these in the few minutes I have. In 1971, in this paper, Lindsay discovered that there are proteins that are packaged inside the phage heads. These are highly positively charged histone-like proteins, which he called internal proteins, IP1, IP2, and IP3. People thought that these might be important for packaging, but in a series of papers, Lindsay showed that actually these proteins are not essential for packaging, but important to protect the released DNA in certain E. coli strains, which are otherwise restrictive to T4 infection, because they contain nucleases that degrade the delivered genome. In this paper in 1973, Lindsay was one of the first people who came up with the idea of a scaffolding core that is essential for virus capsid assembly. Since then, it became a fundamental concept of assembly in all large icosahedral viruses examined. When not much was known about the function of the portal, in this paper, Lindsay was one of the first people to show that the phage portal is important for DNA packaging. He identified cold-sensitive mutants in the portal protein that assembled ProHits normally underwent all the maturation cleavage reactions, but are totally defective for DNA packaging. He also hinted that the mutant portal was defective because it could not engage with the packaging motor. Many years later, as spirited debates were still raging about the function of the portal, and many, if not most people in the field thought that the portal rotates, and this rotation is coupled to DNA packaging, Lindsay came up with a clever and creative idea to tether the portal to capsid through a fibrous protein known as HOG, and showed that the tethered portal packaged DNA normally, essentially putting an end to the portal rotation models. Lindsay graciously allowed me to tease out the packaging machinery of phage T4, though it was not his intended project for me when I joined his laboratory. In fact, I'm very grateful that he even hired me because I did not know anything about phage when I joined his laboratory. I spent many years on this project, and we published the first biochemical papers on the packaging system. Lindsay also published several papers on the application of T4 to biotechnology. Here again, his creative ideas shined. He discovered a capsid targeting sequence and used it to localize foreign proteins in the scaffolding core. Surprisingly, the phage survived and carried these proteins, which can then be used for various biotechnology applications such as vaccines and gene therapy. With biophysicist colleague Chris Ray, Lindsay came up with a novel single molecule fluorescence assay to analyze packaging in real time and published a series of papers analyzing various details of the packaging machine. Perhaps the most time Lindsay spent was on a packaging model he came up with in 1978. It was his favorite model, in which he strongly believed that the packaging substrate, the DNA itself, might be involved in the packaging force generation mechanism. His ideas evolved over the years, but the concept remained. In 1978, in this paper with Silverman, he came up with he proposed that the packaging proteins energetically introduced supercoils into the DNA, and relaxation of the supercoils generated packaging force. More recently, he thought that DNA gets crunched and decrunched, or it oscillates between A and B forms, generating the force. We still do not have a complete answer yet to this question, but this is one thing that he was extremely passionate about, almost obsessed about. And in fact, it was his last paper on this subject just months before he passed. From the sample of publications that I showed, you may have noticed that almost all his papers are with one or two authors. He was never flashy, always maintained a small group of students and postdocs, and just did his thing, focusing on science. His science was always creative, out of the box, and most of the time against the tide. He was also a true bench scientist. He and I shared a bench. For all the years I worked with him, he was on the bench till the very end. Even while he was facing serious difficulties with his health, in December of last year, about a month before he passed, he was giving samples to his colleague, Chris Ray, but no one knew what was going on. In fact, his colleagues came to know of his passage days and weeks after it had happened. Lindsay was an extremely private and shy person, and extremely polite to a fault. Yet, he touched many of us in significant ways and manifested his humanity with just a few words, or just by his mere presence. I will show you a small sample of touching words from our colleagues. Passionate and audacious, think outside the box, boldness to do difficult experiments with novel approaches, drive to do rigorous science, one of the most creative thinkers I have known, tremendously gracious and inviting, passionate interest and deep knowledge, unique ideas and experiments. I was also fortunate to see a slightly different, not so reserved side of him too. In the years I was with Lindsay, three of us, Lindsay, myself, and another postdoc, Arthur Zachary, we used to go to lunch together almost every single day to the historic and famous Lexington Market in Baltimore. Lindsay used to be quite free during these times and engaged in all sorts of discussions, even crazy ideas about science, politics, and so on, with a sense of humor. These were etched as very fond memories in me and will remain forever. Lindsay's passage was shocking and a tremendous loss to the PVA community, but his contributions and ideas will live on forever. Thank you all for allowing me to share this brief tribute to Lindsay Black, who was one of the most respected researchers in phage assembly. Thank you.